Hello everyone. We are live on YouTube. We can start with the session. Thank you for your patience.
Maybe we wait for a minute or so. It's 5:28 in my.
Yes, Moshmi, maybe we can wait for 2 more minutes. Yeah.
Thank you. Hi, everybody. Welcome to the second day of our 5-day workshop series. This series has been organized by Drugs for Neglected Diseases Initiative, DNDi, in collaboration with BRBC Venture Center and Indian Society for Clinical Research, ISCR. The title of the series is Taking Novel Drugs from Lab to Clinic, Concepts and Case Studies. Yesterday, on the first day of our series, many of us heard from experts about lead optimization process, the entry point in novel drug development pathway. Today, we move to the next topic in drug development, that is pharmaceutical development. And we have Dr. Amit Antarkar with us to chair this second webinar session. Dr. Antarkar brings with him 26 years of experience and knowledge in pharmaceutical formulation R&D, both generics as well as NC formulation across various dosage forms for domestic and international markets. Dr. Antarkar is currently heading the oral solid dosage division within Mylan R&D in India. Before joining Mylan, a Viatris company, he had worked in multiple national and international companies like Teva, Actavis, and Watson Cipla. Dr. Antarkar has his PhD in pharmaceutical technology from Mumbai University, and he has more than 15 patents and publications. Before I hand over to the session chair, a small housekeeping announcement. I request all of you to rename your device or Zoom portal with your registered name. That would help us to generate participation certificate. Thank you and welcome again. I welcome Dr. Antarkar. Floor is over to you. Thank you. Amit, you are on mute.
Yeah, thank you. Thank you, Moshmi. Good evening, good afternoon, and good morning to all the participants and our distinguished speakers. I also welcome all the members, who have made this session possible. And today is the second day of our seminar, and which is going to talk about, the formulation development. So we will be covering topics on formulation development today. And basically, all of us know that in the phase 1 clinical studies on new drug, the availability of the suitable dosage form is essential. This dosage form is most likely to be the final dosage form which would get approved after the completion of successful clinical trials. In today's workshop, we will understand the key requirements for developing a pharmaceutical dosage form for our novel APIs. For doing the same, we have some distinguished speakers with us today to speak on the relevant topics. I welcome our speakers. We will be having 3 speakers today with us. Mr. Ravindra Kodipayka, Dr. Vivek Kumar Ravel, and Dr. Stephen Robinson. After the speakers speaking, after they complete their talk and presentation, we will be having question answer session. So I would invite all the participants to send out their questions so that the speakers could reply to them. Our first speaker for today is going to be Mr. Ravindra Kodipayka, who is going to speak on the topic of impact of API physicochemical properties on the drug development. Mr. Ravindra is currently working as a head formulation research services at Origin Pharmaceutical Services, Hyderabad. Mr. Ravindra has 26 years of experience and has expertise in formulation space, in pre-formulation, formulation design and development, technology transfer and project management. He has a deep knowledge and experience in solubility and viability enhancement of poorly soluble molecules. He has designed and developed multiple formulations in the generic and NC space. Before joining the current organization, he has worked with companies like Gufic, Cadila, Suven Life Sciences, Dr. Reddy's. Mr. Ravindra received Master's in Pharmacy from Kakatiya University and has published a number of articles and patents in the field of pharmaceutical research and development. I invite Mr. Ravindra to start his talk on the impact of API physicochemical properties on the drug product development. Welcome, Mr. Ravindra.
Thanks, Amit, for such a nice introduction. Before I get into the topic, I just wanted to thank DNDi team and other sponsors who gave me an opportunity to interact with fellow colleagues and friends who represent pharma community. So with this introduction, I would straight away go into my topic. So the time limit that we have in front of us is 20 to 25 minutes, and then I have a lot of slides to cover. So what I will do is I will first share my screen, then one second, there is some issue. Hope my screen is visible to all of you.
Yes, sir, it is.
Okay, thank you. Thank you very much. So as I mentioned, in order to better utilize time available, I will focus more on certain aspects which are, critical for from product development standpoint. So I will start with drug substance characteristics, and then spend some time on understanding the impact of physicochemical characteristics and their impact on drug product development, and then also try to talk about some of the case studies. These case studies are from our real life experience. So I'm planning to spend more time on introducing or sharing some of these examples more than spending time on concepts and then theory part. So this is how I'm going to take my presentation forward. So to just start with the introduction part. So all of you are familiar with this slide, which talks about various physicochemical properties and the bulk properties of drug substance, which are typically characterized in the beginning of or early phase of the research, discovery research. And in case of clinical program, we expect or we generally anticipate all these characterizations would have been done by them. But still, many a times we see the data is not published or the data is not available. And sometimes we end up redoing all this work. But more important here is to understand what are these physicochemical properties and understand the impact of these physicochemical properties on the product development. So I will try to focus on some of those aspects than trying to talk everything about each of these parameters, because each of these parameters can take at least half an hour if you really want to dwell deep into each of these aspects. So I request the participants to excuse me if I speed up on some of those slides, but happy to answer if you have any questions towards end of the session. Right. So solubility, as you know, is one of the challenge that every formulation development scientist comes across the moment they start working on a project. So that's why I picked up this as a first point, and nothing to talk much about the definition, but it's very important for everyone to be aware that there are three aspects of solubility that we talk about during our research: kinetic solubility, equilibrium solubility, and intrinsic solubility. Each of them carry their own importance in the field of formulation development. So kinetic solubility, as you know, is the solubility that is determined very early in the beginning or very in the beginning of the formulation development, particularly early phase of the clinical development, early phase of the discovery part, like particularly hit to lead identification or lead optimization or candidate selection. So early part of the discovery program, mainly to address some of the issues related to biological assays. So this is a good parameters to have it, but may not represent the true solubility, because equilibrium solubility, which is generated using excess solid in a saturation condition, is the one which is a true representation of the solubility. So the kinetic solubility, as you know, is typically done using DMSO solution. So we might end up overestimating the solubility. So therefore, equilibrium solubility is a real measure of the solubility. Similarly, intrinsic solubility is the solubility of the compound when it is in a neutral state or when it is not in the ionized state. So coming to the solubility, not to explain much about the factors that will have an impact on the solubility. So temperature, ionization behavior, solvent system, the pH selection is also equally important, and salt and its counter ion will have an impact on the solubility of the active substance, particle size, and solute component. So all these things will have an impact on the solubility. So it's very important for a formulation development scientist to be aware about these impacts, so that they can determine right formulation and processing conditions when they start working on designing the formulation or developing the formulation. So solubility is everywhere. So you can see on right side, which is highlighted in yellow. So solubility is a prerequisite at each stage of the development, including the dosage forms which we work upon. So in case of parenteral solutions, without solubilization, you can't develop a injectable liquid solution. So solubility is equally important. And similarly, lipid-based drug delivery systems, we talk about much in subsequent slide. We talk about this much in subsequent slides, but it typically requires drug to be solubilized in oily vehicles. Similarly, topical ointments, they do require API to be solubilized in oils, alcohols, etc. And tablets, capsules, not very critical, but I think many of you would have experienced that the high soluble nature of API doesn't allow a granulation, particularly the wet granulation, because immediately you will see kind of over wetting happening in the system. So therefore, understanding the solubility of API and identifying the right approach is equally important even in solid oral dosage forms. Spray drying, obviously, selection of solvents and then solubility is equally important to develop the right composition, particularly spray dried composition. So I'll move on to the next slide. So determining solubility alone is not enough, because solubility is only talking about one side of the coin. It might address your challenges in the formulation development, but will it address the issues related to the biological systems? So that's where the permeability aspect comes into the picture. So it's very important for all of us to refresh our memory, for those who have already seen these kind of slides, but more important to refresh ourselves on and understand the impact of permeability and solubility on the bioavailability, which is one of the major challenge that we come across in our day-to-day development work. So on the right side of the slide, I have presented a very well-known biological classification system, which is also part of M9 biopharmaceutical classification system, which is an ICH guideline. So it's a typical 2x2 matrix dividing active ingredients into 4 classes, and class 1 represents high soluble and high permeable compounds, and then class 2 low solubility, high permeability, and class 3 high solubility, low permeability, and class 4 is low solubility, low permeability. I'm sure many of you are aware that class 2 and class 4 are the most challenging ones in terms of designing a suitable formulation to achieve required solubility and bioavailability. So while this biopharmaceutical classification system acts as a guide to understand the behavior of the active ingredient and also design a suitable formulation strategy, but it has certain limitations. For example, when we talk about permeability, it is related to the extent of absorption, and unless you know what is the extent of absorption, it's difficult for you to understand or classify your drug molecule into one of these classes. And similarly, you are supposed to run solubility study across the pH range in a buffer system. And many a times in the discovery phase, I don't think the scientist will have time, or scientists will have that luxury of generating a lot of data, particularly because the compound availability is of a challenge. The quantity of the compound available will be in the range of milligrams. So very difficult to run these kind of studies, because saturation solubility studies is also one of the requirements. So to run this saturation solubility as per BCS classification system, we are supposed to run, we are supposed to consume grams of material. So many a times it becomes difficult for scientists to generate data as per BCS classification system, particularly in the early part of the discovery program. So to overcome this, one system that was proposed by Dr. Dressman and the team is DCS, Developability Classification System. I think some of you must be aware about this system. The good thing about this system is that you can apply this system to any situation, and it gives you an idea about the complexity associated with your molecule from the developability standpoint, not just as a regulatory submission standpoint. So what is the difference between the BCS system and the developability classification or DCS system? So DCS, as per DCS system, certain improvements have been brought into the system, and these improvements are as follows. Like the liquid or the solubility, for conducting solubility as per BCS, you are supposed to take 250 ml of the buffer system, whereas in DCS, the quantity that is considered here is 500 ml. These are all done based on a series of experiments, and then understanding that Dressman has come out with, they felt that 500 ml seems to be more pragmatic than restricting it to 250 ml, and that 500 ml, the media that is to be considered is FASISIF, fasted state simulated intestinal fluid in place of buffer system, which is more representative of the in vivo conditions. So these are the changes on solubility front, and on permeability front, there is no compulsion for you to use FABs, which is dose fraction absorbed, which represents the extent of absorption. Here you can always use the data that is generated during discovery phase. So you can use cell line, the data from cell line studies, you can use from in silico predictions. So therefore, at that point of time, with that data, you will also be able to identify and classify the complexity of the molecule in one of these classes. Now, one of the main differences between BCS classification to the DCS classification is that the class 2 compounds have been divided into two parts. A part which is limited by the dissolution, the compounds which which come under dissolution rate limited compounds are categorized or classified as class 2A compounds, and those which are solubility limited are classified under 2B. This is one good development, because many times by looking at low solubility and high permeability, we might end up designing formulation approaches which are very complex in nature. And sometimes those kind of strategies may not be needed, but because it's class 2 compound and you don't want to take a risk of developing a formulation which doesn't meet your bioavailability criteria. So sometimes we end up overdoing in terms of formulation development, the strategy and the development part. But in case of DCS, it allows you to look at the stage and understand whether your absorption is limited by the dissolution or your absorption is limited by the solubility. So those molecules which exhibit high permeability, low solubility, and high permeability acts like a compensatory mechanism for solubility. In those cases, the sick conditions can be met very well. So therefore, you may not require to implement enabling technologies like approaches towards solubilization technologies like nanoparticles, microemulsions. These are some of the complex complexities that we generally venture into when we find a molecule is practically insoluble, and then sometimes ignore that high permeable nature of the molecule also acts like a compensatory to the solubility that thus creating sink conditions in the in vivo systems. So therefore, this system allows you to understand whether it is a real solubility limited or dissolution rate limited. If it is a dissolution rate limited, then we need to understand what is the particle size above which it becomes dissolution rated. So in that case, determining the particle size and then developing a simple immediate release formulation is good enough in case of class 2A compounds. Whereas in class 2B compounds, similar approach may not work because the solubility is so low that the concentration of the drug under in vivo conditions will exceed the solubility of the drug molecule. So therefore, simple technologies like micronization may not help you to achieve required solubility and bioavailability. So therefore, in this case, class 2B are the right candidates for looking at enabling technologies which are linked with solubilization. So class 3 and class 4 are typical examples are typical representatives of class 3 and class 4 of BCS systems. So I will not spend much time. In the interest of time, I will move on to the next slide. But before moving on to the next slide, I would recommend you to go through these articles, which give you a lot of information along with a number of examples in terms of how do we identify, how do we classify, and how do we investigate. Because one of the challenge here is that you are going to take data which is not a representation of human data. Similarly, solubility data that you generate is not representative of the human data. So with these limitations, when we classify the molecule into different classes, there is always possibility that we might end up in a over prediction scenario or under prediction scenario. To minimize the risk of under prediction or over prediction, there are certain investigations that are recommended by Dr. Dressman, and those investigations will have to be carried before you confirm the class of the compound, and because that can have an implication or impact on your development strategy, product development strategy. So my request to all of you is please go through these articles. They give you a lot of information about these things. With this, I will move on to the next slide. Yeah, this slide talks is basically to give you an example wherein we made use of micro dissolution testing apparatus. So as you know, dissolution testing is typically done in a 900 ml dissolution vessel, and compound requirement is also very huge. So in place of normal dissolution apparatus, you know that Payan has come out with a micro dissolution apparatus. So we have made use of this apparatus to understand the differences in different salt forms that we screened during discovery phase of the development. So this graph gives you an idea about what we have exactly done. So there were two salts, hydrochloride salt and sodium salt. We are supposed to select or suggest the best salt from the formulation development standpoint or developability standpoint. And then one of the criteria that will determine which salt to be selected is the solubility. So when we have done a solubility study in a simple water system, we understood that the solubility is much higher in water. Solubility is much higher for sodium salt when compared with hydrochloride salt. But there were some challenges in dealing with sodium salt. There were many handling related challenges that were faced. So then we were stuck in terms of what should be the right approach to select a salt that can go for next preclinical Pka studies. So we have done one dissolution study using micro dissolution apparatus. I think some of you must be knowing this. Micro Dis Profiler is the one online measurement system. So you can run for number of hours, number of minutes, and generate a continuous profile of your molecule to understand the behavior in the dissolution vessel. And you don't need to use grams of quantity. So just 2 to 20 ml with milligram level quantities are sufficient. So this is more suitable for discovery setup, particularly during the stage of lead identification, lead optimization, and candidate selection stages. So what we did is we have run this study, and in addition to the salts, we have also prepared some solid dispersions of the same salts, and then we have run the study using free base with solid dispersion. So from this graph, you can easily make out that the salts, the one which is presented in red represents sodium salt, and the one which is in blue is a HCl salt. Both of them show similar behavior in terms of the dissolution. And of course, there is a precipitation tendency, but the solution is stable at least for more than 100 minutes before it starts showing signs of precipitation. And whereas in case of solid dispersions, you can expect, as the nature of that formulation itself is meant for improving the solubility, so you don't see the precipitation. But here idea was not to select a formulation, but select the suitable salt. So that's where we ended up selecting both of them, and this is done for a customer, and customer did the study, and both of them were found to be suitable for that stage of development. Whereas a single point analysis was not really giving you any clue. So that's what I wanted to highlight here. Hope I'm clear. I'll move on to the next slide.
Ravindra, you have 10 minutes more.
Oh, is it? So I'll quickly run, then I think I've just not even completed 20% of it. So I'll quickly run through the slides. I will not read much. Okay, this is a case study wherein we had a molecule with poor solubility and low bioavailability. And on right side, you can see the solubility data that was generated was not really encouraging. And also, it had a pKa of 3.0 and weakly basic in nature. So you can imagine the challenges associated with this molecule. And solubility was not achieved, and pKa was not favorable. So we ended up, this is giving some characteristic profile, ionization profile of the same molecule. So this one is clearly representing solubility behavior, which is inconsistent with ionization behavior of the molecule. And then we ended up developing a formulation which is an amorphous solid dispersion with an in situ salt formation. So the ones which are given on right side, one is in orange and green. These are tested along with conventional suspension and nano suspension. You can see the target AUC was achieved with a solid dispersion wherein in situ salt formation was done. Okay, this is another case study which is for parenteral route of administration, and again similar solubility issues. It was not soluble in organic solvents, not soluble in lipids, and not soluble in presence of complexing agents, surfactants. pH modification was not helping. So then we ended up developing a nano formulation, nano suspension formulation with a typical, size of D90 in the range of 300 microns. That really helped us to, provide this formulation for conducting preclinical tox study. I'll quickly move on to the dissociation constant of pKa. I will not talk much about the theory part, but very important for all of us to be aware that pKa can have an impact on solubility and permeability. Solubility, because ionization behavior will determine will indirectly influence the solubility of the compound, and permeability, because that permeability is better in a neutral form, in a non-ionized form as compared to ionized form. So therefore, determination of pKa is also equally important, particularly during the discovery phase, to understand the ionization behavior of the molecule, and then accordingly suggest a formulation that will fit for purpose formulation for that phase of the development, but also will give you an idea about what should be the approach towards clinical formulation development. Right. So this is a lot of theoretical stuff, so I will just skip this, but wanted to touch base on these points which are captured or highlighted in yellow. That how do we design a formulation where the pKa or the ionization behavior of the molecule doesn't allow or doesn't give you bioavailability which is desired in the under in vivo systems, because any molecule exhibiting ionization will have a preferential solubility in a particular site of GI tract, and particularly you know that any basic substance will have a better solubility in the acidic regions, which is represented by the stomach, and then once it goes to the basic region, there is a high chances of precipitation happening. So therefore, it is very important to design a formulation that will ensure to keep the drug in the solubility state, in a solution state, and then ensure the availability for absorption. So one of the approach that we take up for this kind of molecules is to take the advantage of creating a micro environment, pH environment, wherein irrespective of the macro environment, you will ensure that the drug is in a solution state. And similarly, sometimes if there is a window of absorption, then gastro-retentive systems can also be thought about, because it will allow you to ensure the drug goes into solution before it reaches the site of absorption, so that it is available in liquid or solution form. Then absorption becomes much easier. And similarly, in parenteral formulation development, understanding ionization behavior is very important, because that will help you to set the right pH conditions to ensure the solubility of the bulk solution. So one more case study, I'm just presenting it here. This is again talking about the impact of pH solubility and stability of the product development. This I picked from my experience from a generic background. But very interesting case study. This is, you can take the name of the molecule. It's a dabigatran. That it's used as an anticoagulant, and then the dosage form that was developed was pellets filled into capsules. So you can see this. It exhibits two pK values, 4.0 and 6.7, and it's a weakly basic in nature. So this pK value is very much reflected in the solubility data. So the solubility between pH 4 and pH 7 is very poor. So therefore, it's very important for us to ensure that the solubility is equally better. Otherwise, the major site of absorption is intestine, where the pH will typically start from a duodenum to jejunum and ileum put together. Say starts from 4 and typically 4 to 8. That is the right pH range to ensure intestinal absorption. So we have developed a system that helped us to ensure microenvironment of the pH, and then that this is a tartaric acid core onto which we loaded drug, and both of them are separated with a polymer layer because of the incompatibility between acid and base, and that helped us to achieve the required bioavailability. So the blue one and the red one are the reference product, and the one which is given in pink is test products. You can see superimposable bioprofile of the formulation. So that's how I think if we understand the pKa, understand the solubility behavior, it will really help us to understand what should be our approach to the product development. Partition coefficient, again, this is also an important parameter, particularly during the discovery phase, when you are screening a lot of compounds. You want to understand whether they have a desirable lipophilicity or not. Otherwise, you end up doing development and optimize the lead, then realize the physicochemical characteristics, particularly the partition coefficient of that molecule, is not favorable for subsequent studies. So it's always advisable to conduct such studies very early in the discovery phase, and this doesn't require too much of an investment. So therefore, as a preclinical formulation development scientist, we are supposed to run these studies. And N-octanol water system is an age-old technique, which will give you an idea about the log P value. Now, how do we understand this log P value? So particularly, if the value is in the range of 0 to 3, then we say it's an optimal, because it exhibits both hydrophilic and lipophilic behavior, which is suitable for ensuring solubility, and then also helps the molecule to cross the biological membrane when given under in vivo conditions. But anything that is less than 0 is indication of highly hydrophilic in nature. So there will be a challenge in dealing with these molecules because very difficult for them to cross the biological membrane. So similarly, those molecules which exhibit a value more than 3, particularly in the range of 5. The another challenge with these kind of molecules is that how do you design these molecules, or how do you design a formulation for these molecules because of highly lipophilic in nature. So some of those examples from the literature, isotretinoin, butasteroid, these are all highly lipophilic substances with a log P value of more than 5. So one of the solution could be lipid-based drug delivery systems, and I will not go into these details. But by using lipid-based drug delivery system, you can always take the advantage of lymphatic delivery, and that's how you can enhance the bioavailability. Of course, this is just an example. I'll skip this slide. And then one more case study. I will not talk much about it, but this is a case study which is relating with a molecule which exhibits a log P value of 6.8. So the best approach was to use an emulsion technology, and then we were successful in doing so. We have developed a SMED system, and then we were able to achieve required solubility and also the stability part, which was very critical in this particular molecule. The stability was very poor. It was showing all kinds of challenges with oxidation, photodegradation, moisture-related hydrolysis, thermal degradation. With this emulsion technology, we were able to improve the stability profile. We were not able to address the problem from the root cause, but we were able to improve the stability characteristics of the molecule. And the required dissolution was also achieved. And then stability was much better. Even after 6 months, we could see the total impurities were not crossing even 0.5%. So this is the slide which talks about the stability part. And as you know, stability is also one of the challenges that we encounter as part of the drug product development. And then these are common degradation pathways. So understanding about these pathways and relating them with the molecule, understanding the chemistry of the molecule is very helpful, particularly in the beginning, early phase of the development, because there will not be much data available. So you may want to run one forced degradation study, or you may want to predict the degradation pathways or behavior of the molecule based on the chemistry part. So those are the aspects that we need to look into as a formulator. But my intent here is to take you some examples from the product development standpoint, because I will not be able to spend time on each of these things. In the interest of time, I will just skip this slide. Okay. So
Ravindra, you have 5 more minutes, and then there is going to be some question answers. So
sure. I will try to finish in 5 minutes. So hydrolysis is one of the common challenge degradation that we encounter in the formulation development. Aspirin is a classic example. So I will not go into these again. I will talk about some of the important points that we need to take into account while developing a formulation of a molecule which exhibits hydrolytic degradation. Simple things that we consider is, try to avoid hygroscopic excipients, and then as far as possible, try to select a process which is dry, like for example, direct blending, dry granulation. And packaging, that it's a regular practice to use desiccants to avoid moisture permeation or to minimize moisture ingress into the drug product. And multiple systems are available, silica gel systems, and then molecular sieves. So we need to run these packaging studies to make sure that the formulation is not only the formulation part, but also packaging provides adequate protection. Okay, understanding pH conditions, providing moisture barrier coatings, These are all common things that we come across. And the use of moisture scavenging or sequestering excipients such as New Zealand starch 1500. Sometimes we think that these are all hygroscopic materials, so particularly starch 1500. But we assume that these are all hygroscopic in nature, so they are not recommended for use with some of these active agents. But our experience says, I have an experience dealing with one immunosuppressant. I was avoiding the usage of disintegrant, which is cross-povidone, because of its hygroscopic nature and water absorbing capacity. So then we realized that use of colidone was really helping. What it does is it will preferentially pick the moisture from the surrounding environment and keep the molecule dry, or active molecule in a relatively dry state. So that was really helping in certain cases. So sometimes it is very important to understand these aspects and then do a proper selection of the excipients. Oxidation, I will not, I don't, I'll just keep this. So I'll go to this yellow part of the slide. Controlling metal ions, peroxides, addressing dilution effects. Sometimes concentrated solutions give you a better protection. Determining optimum pH range, because pH can also aggravate the oxidation. So therefore, understanding the impact of pH on oxidation is very, very important. And packaging selection, there are a lot of choice available in terms of which packaging to be selected. So in this particular case, we should select we should select those packaging material which are less permeable to oxygen. For example, generally we end up selecting a blister packaging material which is alu to protect it from moisture. But many of you must be knowing that PVA could be a good oxygen barrier. So therefore, if the degradation is triggered by oxidation, not by hygroscopic behavior or moisture permeation, then better to select PVC, PVDC kind of pack, which will provide a better protection compared to alu blister pack. Similarly, use of antioxidants with a redox potential which is lower than the active ingredient, so that they preferentially undergo oxidation and protect the drug substance in the drug product part. Thermal stability is not much to talk about, because there is we don't get much leeway in terms of addressing the problem using formulation approaches, because thermal stability is an inherent property of the molecule. But however, we can make sure that the formulation part is taken care in terms of excipients, the process, so that the thermal degradation is not aggravated by the adjacencies, particularly the incompatibility with the excipients, or no presence of moisture or water. Those are the things that we need to take care in thermal stability.
Ravindra,
Photolysis, I'll just skip this slide.
Ravindra, your time is over. So
Okay.
we will maybe in a minute, if you can wrap up so that we can take questions.
Sure, 2 minutes. Give me 2 minutes. I'll
Sure.
try to close this.
Sure. Thank you.
Yeah, and these are the case studies I wanted to share with all of you, which talk about the stability, which highlight the stability part. But in the interest of the time, I will switch over quickly to polymorphism. And this is an interesting topic. Of course, it requires a lot of time, and unfortunately, we are left with only 2 minutes. But just to let you know that all these aspects of polymorphism are critical in the formulation development, because interconversions between polymorph to other polymorph, what we call as an anisotropy, and between amorphous to crystalline forms are some of those challenges that we encounter. So understanding the polymorphic behavior is very, very crystallinity and polymorphic behavior is very important as a formulation development scientist. So generally we use XRD technique as a screening technique, but there are other techniques which can be used if your level of detection demands a much better sensitive equipment or instrument. So I'm just summarized some of those things. Sorry. Here, but the point I wanted to highlight here is what are those properties and what is the impact and what are the strategies that we would prefer to select under such scenarios. I'm sorry, I'm unable to spend time, but these are self-explanatory. I'm only trying to understand the impact and trying to identify what is the suitable technology or technique or approach towards formulation development. So some of the case studies I'm skipping, very interesting case studies, but in the interest of the time, yeah, this is one case study. I just spent few seconds here where simple XRD technique was not helping identifying as traces of form 2, contaminated in form 1, which is for clopidogrel. So our team, with the help of Purdue University, developed a method called SSG method, which is an optical method, second harmonic generation imaging, and that helped us to identify form 2 at a ppm level. So always XRD is not the only go-to instrument or technique or technology. The method. Sometimes you want to club with some methods which provide much sensitive, which show more sensitivity towards the analyte that you want to analyze. So sometimes you combine it with Raman. Sometimes come out with hard stage microscopy is also one of the interesting tool for understanding phase transition. So we need to understand the requirement and accordingly select a suitable technique to understand the crystallinity and polymorphism in the drug product. Yeah. So I'll just skip this particle size and effect of particle size. Basically, we try to micronize the materials, particularly those molecules which come under class 2A, and that is good enough for you to achieve required solubility, dissolution, and bioavailability. So majority of the molecules that we deal with are from class 2, and class 2 molecules might require a micronization as a prerequisite. How do we deal with micronization issues? And then how do we deal with hygroscopicity behavior? How do we generate the devious isotherm? So all these are equally important for a scientist to understand the impact of these properties on the product development. So I think these are some of the case studies. I'll just conclude here. The time.
Yeah.
I really apologize for not able to keep time because this topic is such a big topic that we find it difficult to stop somewhere. But I take this opportunity to thank all of you for your patient listening, and then look forward hearing your feedback in the form of questions. I will try my best to respond to those questions. Thank you very much.
Yeah. Thank you, Ravindra. It was a very elaborate presentation, and I definitely agree with you that this topic is such that a time of 30 minutes is really not sufficient, and you had some very interesting case studies to show to the participant. Definitely, I know that participants have a lot many questions to ask you based on your experience. However, we will not be able to take a lot many questions. We will just take one question. Which probably is a question is like, there is a question which was asked is like the role of excipient in governing the solubility and how would that impact the development?
Right. So I think we touched based on this point when I was presenting a case study on dabigatran. So the dabigatran, as I mentioned, is a weak base with pK values which are not favorable for solubility under in vivo conditions. So in those situations, the best way is to convert that pH dependent molecule into pH independent system. That was done by using tartaric acid, which is a well-known organic acid. So tartaric acid was able to solubilize the drug because of high acidic microenvironment, and then that solution was helping in terms of ensuring absorption throughout intestine, GI tract.
Okay. Thank you very much. I think so. It is something which really you stressed upon that the role of excipient can influence the solubility and the bioavailability. So selection of excipient becomes quite critical in the development aspect. Thank you, Ravindra, and to all the participants who have put a lot many questions. Probably you can get in touch with us to help you out to answer any of these questions. Post this session, some email address are there where you can get, and we will try to respond to many of them as possible. Thank you, Ravindra. We move on to our next speaker, Dr. Vivek Kumar Ravel, whose topic for the lecture today is going to be Formulation Strategies for Accelerating Oral Drug Candidates for Phase 1 Clinical Trials. Dr. Vivek is a senior group leader in the Pharmaceutical Development Department of Syngene. He holds a PhD in Pharmaceutical Sciences from MS University, Baroda, and he has over 16 years of experience in formulation development. He has worked in companies like Sun Pharma, Ranbaxy, Novartis before joining Syngene. And I invite Dr. Vivek to present formulation strategies for accelerating oral drugs of candidates to phase 1 clinical trial. Welcome, Dr. Vivek.
Am I audible to everyone?
Yeah.
Okay, thank you. First of all, thank you, Dr. Amit, for the nice introduction. Thank you, Dr. Ravindra, for setting up the platform for my talk. I think you made things easy by setting the path. And also thank DNDi for selecting me as one of the speakers for this seminar, and all the participants for taking out time and coming here to join this meeting. So today my topic would be to see what can be the formulation strategies and how we can accelerate the oral drug candidates and then hit the first human clinical trial. So as we all know, so as formulators, every day we are facing the type of compounds which we are getting for development. They have one or the other issues, I would say, or specific characteristics. So they are either low soluble, they have limited permeability, or they have processing challenges like poor flow, or poor compaction properties which cannot be converted into tablets and so on. So one such example is that there is a group of compounds called the PROTACs, which is like a recent set of, or a class of compounds which is being widely explored for cancer therapy. And then these PROTAC compounds have a very specific set of characteristics, like they have very high molecular weight, they have a labile peptide bond which makes them very chemically unstable. They have very poor cell permeability, low solubility, and they're highly potent. So by low potency, I mean that their doses will be very less. And so making low dose compounds, ensuring content uniformity unit to unit will have its own challenges. So just wanted to give you this example to show that what are the challenges which we face every day as a formulator. And these are the type of compounds we handle day to day. And as we know that there are a lot of discovery compounds which actually move into into development, and they move through a lot of filters of efficacy, the physical properties, the safety, and they finally get the formulator receives it for development. Yeah. So I would just like to stress on this slide. So why are we all here, and what is the ultimate goal? So as pharmacists and formulators and chemists, pharmacologists, and as people in the medical care field, it's our responsibility to make sure that the right drug reaches the right patient at the right dose via the right route, oral or parenteral or topical, and at the right time. Because time, the sooner the dosage form reaches the patient, you have a high probability of saving a person's life. So time is also very important. as equally to the science and all the other characteristics which go along with this. So this is also the US FDA's goal or mission, to have all these right, so that the patients are benefited and lives are saved globally. And we all play a very vital role in making this happen. Yeah. So how do we make this happen now? I would just like to give an overview of how different companies approach. So how can we hit them? How can we make the first formulation in humans? So there are two different approaches. One is called the exploratory formulation approach, or one can be the commercial formulation approach. So just consider a person running a marathon. So either he runs fast in the beginning and then he slows down towards the fag end. So that approach is the exploratory formulation approach, where there are the pace at which the work happens is very fast in the beginning. You are able to screen out a lot of compounds, and you are able to get a lot of information. However, when you move into the later phases of clinical study, your speed will slow down, because these formulations may either be physically unstable or chemically unstable. Your manufacturing process may not be appropriate for commercial scale, but it gives an advantage of speed and agility. Yes, so that's one approach. Think of the second approach, where you go slow and then you can ramp up your speed towards the end. So you make a sort of the close to the market formulation from the beginning. You put a lot of resources in developing. In this way, you're actually able to make a very robust formulation. And by this way, you're actually able to ramp up the speed of your phase 2, phase 3, and as you move to commercial. So, and the risk of this approach is also lower, because you have already studied the formulation to a great extent. So the confidence levels are high. And of course, this each approach comes with its both advantages and disadvantages. So if you ask me what is most commonly used, so most commonly used approach is the exploratory formulation approach, which we will be discussing more in detail in the coming slides. This is mainly when you have a scaffold of compounds, or you have 3-4 targets. You have already seen some variability in your preclinical model. You are not so confident on moving very, very fast on the other clinical, not too confident on your hitting the clinical target. So this is sort of a reserved and conservative approach, I would say, where you don't spend too much of time and money on the phase 1 formulation development. Yeah. So where do we start off with? So there is a lot of information. So when the formulator actually receives the compound for development, he actually has a lot of information from the discovery team. And the formulator actually has to use this as a bridge. He has to go through the physicochemical properties, the biopharmaceutical properties, and use all the information and develop the formulation for the first in human trials. Yeah. So this slide, Dr. Ravindra briefly spoke in his speech. I will just maybe refresh it here. So as a formulator, so what would be our starting point? What is the right formulation approach? Where should we start with? So it's like someone who's standing and he's like looking at which direction to go, right? So there's a scientific rationale on how you can select your first formulation, or based on whatever permeability data and the solubility data you have. So what you have to do is you just have to plot your dose. The dose is the maximum human dose. For example, if your maximum human dose is 5 milligram, and if it is soluble in 100 ml of, so in this case it's FASIF, that is fasting simulated intestinal fluid. So you just take the fluid where the drug is supposed to be absorbed, the site of absorption. So in this case, we are assuming that it is absorbed from the intestinal fluid. So for 5 ml, if it is getting dissolved in 100 ml, so you just have to plot it here somewhere around the 20 mark in this scale. And you have the permeability data from the CACO2 cell lines. So for example, if it falls somewhere around this area, so it falls under the DCS class 2B. So there are some guidelines. So when it's class 2B, then they are water insoluble and they are solubility limited. So in this case, the preferred approach is to use lipid-based systems where you dissolve your drug in some lipidic excipients, and there's a higher probability of you increasing the bioavailability in animals or in human models. So similarly, if your compound falls in 2A, you can go with a micronization approach because it is just solubility rate dependent. If you are able to ramp up the rate of dissolution, you have a higher probability of hitting your increasing your bioavailability. So similarly, if your compound is class 2 or class 3 or class 4, that means the permeability is very low, and the only way is you have to use a combination of lipid carriers as well as efflux protein inhibitors. So some of these drugs can be efflux protein substrates. So our intestinal lumen is rich in efflux proteins. So these proteins actually identify your drug, and they actually do not allow the drug to move from the lumen to the blood. So these proteins can be inhibited by some efflux protein excipients, for example, poloxamers, polysorbates. So these are some reported efflux protein inhibitor excipients. So you use your drug along with these excipients in a lipid carrier, and you have a higher probability of increasing your exposure in animal and then in humans as well. Yeah. So moving on to what are the key considerations which you should take as a formulator when you actually consider your formulation development. So there are, I've just put this into 6 buckets, and I'll go to each one. So first one is the physicochemical properties. So most of these were partially covered by Dr. Ravindra, but yeah, I'll quickly take you through these ones as well here. So first one is understanding the physical stability of the drug substance. So it's important you know that the polymorphism, does the drug exhibit polymorphism? This is very important when the drug is having low water solubility, and if different polymorphs have different stability as well as solubility. So you have to understand this, and you have to ensure in such cases, to the best possible extent, that the polymorph doesn't convert over stability, and also during your manufacturing process. In case you are doing a wet granulation, and when exposure to the aqueous media, if there's some polymorph change, these are some considerations you should have while developing your formulation. So understanding physical stability is a very important criteria. Chemical stability, yes, it's important you understand under which conditions your drug degrade. Does your drug degrade in the presence of water? So you avoid excipients which have higher moisture content. There are excipients which are available nowadays with lower moisture content. You select your excipients appropriately. Is the drug prone to oxidation? You can use an antioxidant. Is the drug light sensitive? Then you have to manufacture the drug in a light sensitive manufacturing area. Is the drug heat sensitive? You process your formulation by not applying too much of heat. So very important that you understand under which conditions your drug degrade before you start your formulation. Yeah. One more key aspect, particularly for solution dosage forms, is understanding the pH at which the drug is stable. Is it pH sensitive, with respect to solubility as well as stability. Plus, is it ion sensitive? So there's a warfarin sodium is one of those molecules which is like being in the market for many years now. So it's very sensitive to ions in the media. So if it encounters ions, pH, if it's more stable in purified water when compared to a water solution with some sodium salts. So you understand whether your drug is ion sensitive, so you can avoid such sort of incompatible excipients as you develop the formulation. With that, I'll just give one case study which we were fortunate to work on. So just to give a background of the molecule, so the molecule exhibited two forms. We called it the form 1 and the form 2. And fortunately, these two forms had the same extent of solubility as well as stability. So that was one less headache for the formulator. And for early phase, we decided to go ahead with either of the forms because speed was the essence, and that was a clear-cut requirement, but not at the expense of not understanding the differences in form. So we figured out that what are the differences between the two forms, and we took a scientific rationale on moving ahead. We understood the pH-dependent solubility. So this compound had more solubility in the acid phase, and practically insoluble in as it was beyond pH 2 and beyond. So this required a clear-cut enabling technique to actually, it had to be solubilized by using some external components. So how we did this was the way. So this first-in-human composition was given as a 2-bottle configuration. So one bottle had the co-solvent mixture. The co-solvent was actually beta-cyclodextrin. It was a beta-cyclodextrin solution. That was one. And the second bottle had the drug powder. And at the clinic, where the drug is getting administered to the patient, the medical practitioner has to actually open this bottle. He has to add it into the bottle number 2, having the drug powder, sonicate it for 5 to 7 minutes, and he gets a solution of drug which has 20 mg per ml. And this is further dosed into patients. Why this approach? By this approach, we were able to quickly hit the market because it was just plain drug into bottle. We could get max, there were no excipients involved. So we could quickly move into the market by minimum experiments as a formulator. And why we had to give two different bottles was the solution stability was again a concern. We couldn't have a ready-to-use suspension, so we actually had a compounded suspension at the clinic. So this was all to facilitate that we hit the first-in-human trial quickly. We get phase 1 clinical readouts faster, and we move quickly in the clinical program. So these are common approaches which are now being frequently adopted by many companies to accelerate the phase 1 or the first-in-human clinical trials. So some key takeaways from the compound was so it definitely needed solubility enhancement. There were 2 forms available. Fortunately, they did not have differences in solubility. Co-solvent and cyclodextrin complexation approach was taken. And one more advantage I missed to mention is this was the same formulation which was used for doing the tox study in animal models. So we could extrapolate the same tox formulation into humans. So this is sort of we could really accelerate this program into the humans for the human study. The drug substance also, we were able to ramp up the manufacturing, the desired form, and the impurities were within the desired limits. And the drug in bottle with 2 separate bottles was the quickest format we could support for the clinical study. Moving on to the next critical point is powder properties. So powder properties. So particularly when it comes to, again, poorly soluble compounds, it can be particle size can be very critical, because larger the particle, I think the solubility is again going to be even bad for when it's poorly soluble, or even wettable in that case, if it has wettability issue. And again, particle shape is very important when the flow of the powder is poor. I will just give a real-time example of one of the interesting case studies we had. So there was a compound which was actually rod-shaped. So there were multiple clinical trials we had done with the rod-shaped API. The powder did not have any flow property issues. The blend in which it was actually blended and compressed into tablets was flowing like butter, I would say. It was very, very good. But we suddenly encountered a batch where suddenly the flow was actually stagnant. So there was no flow. So then we actually went, and when we actually studied the API particle morphology, we found out that the API is actually not rod shaped now. It was needle shaped. So then we understood that it's the needle which gets formed first, and the needles grow and form rods. So in this particular batch, somehow the crystallization step was stopped maybe a few hours earlier, and it precipitated out as a needle form, and the needles actually stopped. So just imagine when you have a lot of needles, will they won't flow, they'll actually get interlinked and they'll just stock. So that exactly happened, and this was one of the learnings we had. So particle shape is very, very important when it comes to compounds which can have flow property issues. So sometimes we learn on the way, sometimes we can actually give heads up to our API team who's synthesizing the API to be very critical, give them a requirement that the API has to be rod shaped. Give them a specification. This is an important criteria for me to have the flow. So these are some of the learnings you incorporate as you move forward. Yeah. So that was on drug substance, particle size distribution and particle shape. Going on to hygroscopicity. So it's very important you understand whether your compound has a tendency to take moisture. So you actually expose your compound to different humidities and you check whether it takes moisture. So there's an absorption and desorption curve which you can actually generate, and you can then based on the readout, you can claim it as hygroscopic, non-hygroscopic, or sort of a medium case, in between case also in some cases. Based on data.
Yeah, 5 minutes more for your presentation.
Okay, sure. Sorry. So that's on the hygroscopicity. So important to know, because you will keep your environmental conditions, the right packaging configuration, and the storage condition when you know that your compound picks up moisture. Bulk density is important because when you are actually filling the neat drug into capsules or bottles, you need to know how much volume it actually fills in. So bulk density is very important. Wettability of the compound again is important because selection of the right wetting agent will get influenced by that. One more case study here. Again, I'll maybe only concentrate on the drug development part, which we did. So this was a low dose compound. The doses for the clinical study were 0.5 milligram, 1 milligram, and 5 milligram. And since it was a low dose drug, we had to fix around particle size. We sort of had an agreement with the API team to have particles D90 around 50 micrometers. Sorry. We carried out drug excipient compatibility to make sure the excipients are compatible with the drug. We knew that the drug degrades in alkaline condition, so we had a acidic microenvironmental pH. Since it was low dose drug, we had geometric mixing to ensure the drug is uniformly blended in the blend for final blend for compression. And dry granulation approach was taken because aqueous, there's a probability of solid state change. So we had common blend for 2 strengths and a separate blend for the 3rd strength. And we also made 2 matching placebos. So key takeaways, like we understood that the drug degrades in alkaline pH. So we added the acid in the formulation. We know that it forms changes form when in contact with water. We used a dry process, and manufacturing process also was based on geometric mixing. Moving on to selection of excipients. So again here, a general philosophy which companies normally adopt for first in human study is we always start with the commonly used excipients. There only when the need arises to use a very novel excipient, we will use it. But we'll try to exhaust whatever form excipients which are already there in the shelf or there in the inventory or widely used for clinical studies. Yeah. And how do we do that? So we first check by keeping binary mixtures of your drug and the excipient in different stability conditions. You check whether they are chemically compatible. If they are not compatible, we will either not use it or use it with some precautions. And also the drug load in the formulation, like in a tablet of 100 milligram, if a drug is already 50%, then you need to use excipients which have very good compressibility, which can help in the compressibility of a tablet dosage form and so on. So drug load also plays a vital role, and tablet size also needs to be kept in control. And some other key characteristics are like the surface active agents. In case your drug is DCS class 2A, then where you use your surface, just you have to wet the compound to increase the wettability and the solubility. So these are other excipients and commonly used in case needed. So need basis, you will have to use some special excipients in the formulation.
Vivek, time is up.
Yeah. Maybe I'll quickly just please give me just 2 to 3 minutes. I'll just stress on the stability study and 1 or 2 more points. Okay. So now, since we have developed the formulation, it's important that you stress the formulation to different stability studies. So you have 3-4 formulations, you stress them to different conditions, and you check which is the most stable formulation, and you take that formulation forward. So one requirement is stability, one is meeting dissolution criteria, and one is the right quick manufacturing process. Yeah. So these are some advantages and disadvantages of different approaches. Each comes with an advantage and disadvantage. It is up to the formulator to select the right, whether it's right to go with a solution or a capsule or just a powder in a bottle or whatever. So in the interest of time, I'm just moving a little bit quickly here. So some key learnings or some food for thought I will leave for the team here is understand the drug properties from the developability assessment team, take clues out of it and apply it in your formulation development work. It's very important that you understand the solubility, permeability, the physicochemical properties, and use it in your formulation development. Yeah. And of course, keep your CFT team, the quality and the regulatory team in hand with you, and make best use of the team you have in hand. And the whole goal should be to hit the market, hit the first in human trial as soon as possible.
Thank you.
I'll thank you for this opportunity and patient listening. Happy to take questions.
Yeah. Thank you, Vivek. I think so it was a very, very interesting, presentation covering the aspects of formulation and the, opportunities available to accelerate the development. We have few questions here. One of the question which is, put by a participant is the timelines. What are the typical timelines for developing a FIH formulation?
Yeah. So yeah, thanks for that question. So it's typically around 8 to 9 months. I'm considering starting from the polymorph screening to the initial developability assessment and the first in human formulation development, 3 months stability data and clinical supplement. So roughly 8 to 9 months. If it's an integrated CMC where the chemical substance, drug product, everyone is working in tandem under the same campus.
Right. Okay. Thank you. And a few more questions in the same line is like, what is the batch size for a clinical study in this FIH formulation development?
Yeah. So it depends on multiple factors. Like we need to know how much is the clinical demand, what is the size of the stability program which is going to be done. Is it going to be a full blown out ICH stability, or it's going to be a bracketed approach? What is the amount of sample required for release, retention? So typically it can be as low as 2,000. We have made as low as 2,000, and we have also made a size of 20 to 25,000. So it depends on all these factors.
Do you apply QBD right at the start for these clinical batches, or this is taken up at a later stage of development?
Yeah. So no, at this stage, no. When the essence is to hit the market ASAP. So QBD maybe we'll reserve at this point of time. Yeah. Once we have positive phase 1 readouts, is then we will invest more into the QBD.
Thank you, Vivek. I think so. We have taken 3 questions. There are a few more questions in the general chat. The chat. If you can look at them and probably answer, it would be helpful for the participants. Yeah. Thank you, Vivek, once again, and thank you. And we will move on to our next speaker, Dr. Stephen Robinson. Steve is going to talk on quality requirements for phase 1 investigation products. Steve is working with as a pharmaceutical development director with DNDi. He joined DNDi in May of 2012 with the responsibility for all pharmaceutical developmental activities. Dr. Steve has 20 years plus experience in the pharmaceutical industry in the R&D, manufacturing, and the CMC project and portfolio. Prior to joining DNDi, he held several senior positions with Pfizer's worldwide R&D. Dr. Stephen holds a PhD in physical, organic, and peptide chemistry from the Open University and has an MBA from the University of Kent. He is a chartered chemist and fellow of Royal Society of Chemistry in UK. I welcome Dr. Stephen to start his presentation. Welcome, Dr. Stephen.
Okay, thank you, Amit. I hope everybody can hear me okay. Good evening to those of you in India. Good afternoon, good morning to those of you in other parts of the world. I have to say it's very impressive to see so many people attending the webinar today. And also thank you, Amit, for your kind introduction. Just see if I can move the slide on, which is not moving. Okay, so what I'm going to talk about today, and complementing the last 2 talks, I'm going to talk about quality requirements for phase 1 investigational products. I'm going to talk about the evolution of product and process knowledge during drug development. I'm going to examine some phase-dependent differences for investigational products, and I'm going to consider some key quality requirements for phase 1 investigational products, focusing on control of impurities, analytical method validation, and stability assessment. As this is part of a webinar on translational development, I'll be talking about new chemical entities and first time in human studies, rather than phase 1 studies for registered or generic medicines. So I thought it would be helpful, firstly, to help answer the question, what is pharmaceutical development, for non-experts today. Pharmaceutical development is the process by which a molecule is transferred into a new drug product of suitable quality for market authorization. Market authorization obviously allows a distribution of the drug to the patients. It involves the design, development, and scale-up of manufacturing process for drug substances, as well as the development and drug products, as well as the development of quality control tests and conduct of stability studies. Turning to design, you probably already heard about some aspects of this from the previous speakers, but the design is normally guided by target product profiles that consider the patient needs, the routes of administration, what are acceptable dosage forms, stability, cost of goods, regulatory guidelines, etc. During pharmaceutical development, we make drug substances and drug products at all stages for both non-clinical and clinical studies. And you should all be familiar that this is proceeds according to good manufacturing practice, or GMP standards. And finally, pharmaceutical development, you'll often hear the abbreviation CMC used. It's sort of interchangeable, which stands for chemistry, manufacturing, and controls. So my question is, where and how does pharmaceutical development fit into the overall drug development process? Well, along the top of this slide are the classical stages of drug development, starting with drug discovery on the left, moving through preclinical research and clinical research. Often clinical research refers to phase 1 and phase 2A studies, until we reach a proof of concept for our candidate drug. Then we move into late stage clinical development, which is typically phase 2B and 3 studies before we reach approval and launch. And of course, if we get successful approval, then we move into the commercial manufacturing and marketing phases. Well, first thing I wanted to point out was, as we move from left to right, the quantities of APIs and drug products we make increases. So on the left, we might be making in drug discovery a few tens of milligrams. As we move into preclinical research, maybe 10 or 100 grams, maybe a kilo. And then as we move through clinical research, we start to make even bigger quantities. And in fact, depending on your drug, you may be making hundreds of kilos, and maybe commercially even making several tons. Typically for the pre-POC stage, this would use between about 5 and 10 kilograms of API. Now, moving to the formulations for a moment, you'll see that there are different stages of formulation, and really the different approaches reflect the different aims of the different stages. So for example, a tox formulation gets developed because you want to achieve high exposures in animals. It again have to be very simple and flexible formulations, but often can use some excipients that perhaps would not be used in humans. For the clinical formulation, of course, speed, ease of use, and flexibility of dose is important. And I think developing commercial drug products should be self-explanatory. And we shouldn't either forget that even when the drug development process is finished, we also have to think about post-approval support. So what I'd like to do is quickly introduce 2 concepts to you. And the first one is that you can think about drug development in 2 stages. And the first one is what we call learn and confirm. So prior to POC, we're still actually learning a lot about the drug, how it behaves in animals and human beings. At this stage, the manufacturing processes and formulations are not final. And there's also a very high risk of attrition. So you realize that perhaps 70 or 80% of drugs never really get all the way past proof of concept. Proof of concept, however, represents really a point of inflection for investment. This is where pharma companies make big investments, because phase 2B and phase 3 studies are very large studies to confirm the dose, the efficacy, and the safety of the molecule, and to allow its registration. But also at the same time, we're doing scale up and commercial manufacturing. And this is basically needed in the dossier to show that our manufacturing processes are under good control and give good quality products at the end of it. The second concept is ready by phase 3. And there's a regulatory expectation that the late stage pivotal clinical studies use the intended commercial manufacturing processes and the market product. And normally we make a switch from the clinical formulation to the market product shortly before or around the time of proof of concept. And often we need to perform perhaps some additional clinical studies, of BA or BE studies, to affect that change. But having made that change, we can then be relatively confident that if we use the commercial drug product in phase 3, that the actual market product itself will be approved at the end of this process. So the question is, how do investigational products differ by phase? And actually the answer to this question is that the differences in the types of products are driven by the aims of each of the development phases. And today we're focusing on the translational phase and phase 1. So if we try and compare phase 1 with the later stages of development, you'll see a couple of things. First of all, we're talking about some very small clinical studies, often less than 100 subjects, often less than 50 subjects. Trial durations can be between 1 and 14 days. So the first time in human study is typically a single dose study, for example. Of course, one difference is we do actually have a very wide dose range. So starting from the maximum recommended starting dose through to the maximum tolerated dose in humans, and the dosage forms need to be able to handle this. The investigational product at this stage is typically not a commercial one. And I think Vivek mentioned it has to be simple and flexible. And typically at this stage, we're talking about API in a bottle or a capsule, or maybe a simple powder blend. Manufacturing processes are non-commercial, very small scale. One big difference here, and I think the speakers mentioned it earlier, is that extemporaneous preparations are used frequently in phase 1 studies. Some of you may also know this by the expression compounding. Storage conditions, often we're working at a single clinical site or maybe a limited number of clinical sites. So we have the ability perhaps to store formulations refrigerated or controlled room temperature, which is different to later stage studies, which may be in very hot and humid countries. And stability studies, again, phase 1 is relatively short duration, maybe a year, 2 years maximum. So we don't need 5 years expiry, for example. And of course, if we're using extemporaneous preparations, then we can show very limited stability on the product in use. Maybe less than 24 hours is all that is required. So what does all this mean for quality? Well, quality principles, of course, apply throughout product development, but regulators accept that certain requirements can't be fulfilled completely during the early stages of development. For example, manufacturing processes are not fully developed or validated. The investigational product may be intended only for phase 1, or is an initial prototype that may never be used again, or it may even be abandoned if it's unsuccessful. We have limited numbers of batches, sometimes only 1. And at the time of the first clinical trial application, we would have very limited stability data, maybe less than 3 months. And for these reasons, quality requirements and practices get adapted for phase 1 investigational products. So let's turn to some specific examples, turning first to the control of impurities. So developing impurity specifications is a holistic exercise, and we can consider a number of factors. Of course, firstly, we can talk about the manufacturing capability and what our experience is here from development studies and looking at the results from batch analyses. But remember, at this stage, the number of batches is limited and the process is non-commercial and may not even be the final process. We can control also the input materials into a manufacturing process, but we still have limited experience in phase 1. For example, the final starting materials or the suppliers of those starting materials may not be yet established. We also have to think about the variability of analytical methods when developing limits, but again, the methods may not be optimized yet. And probably the most important parameter at this stage is the qualified level of an impurity. So I'm just going to take a few minutes to discuss how the qualified level of an impurity is established. So what is impurity qualification? Well, the ICH definition is as follows. It's the process of acquiring and evaluating data that establishes the biological safety of an impurity at the level specified. The qualified level can be established a number of ways: through the literature, through chemical safety databases, and through non-clinical studies. And also you have to sometimes consider the patient population and the dosing regimen. Well, in fact, for the most part, when impurities are present in batches used in the GLP non-clinical studies, it's actually quite easy to calculate the qualified level of an impurity. And it's simply this formula on the slide where you take the no observed adverse effect level and relate it to the concentration of the impurity exposed in the tox study. Here, the expected maximum daily dose refers to the maximum daily dose you would expect in clinical trials. Well, there are a couple of ICH guidelines around that talk about impurities, but firstly, Q3A applies to API and Q3B applies to drug products. But I want to really stress here that these ICH guidelines really apply at time of registration. They were never really developed or intended to be used in the very early stages of development. And here's a figure which I've cut and pasted from the ICH Q3B guideline for new drug products. Now, you can see it's a very complicated figure, and I'm not even going to attempt to explain it. But you can see the difficulty of applying the ICH guideline at this stage. First of all, you've got very complex set of thresholds where you report or identify or qualify impurities. But actually, realistically, at this date, you don't actually know the maximum daily dose to apply at this stage. After all, one of the points of phase 1 is to escalate the dose and to find the maximum tolerated dose in humans. So, and in fact, for this reason, there is acceptance that higher reporting, identification, and qualification thresholds are acceptable for phase 1 clinical trials. And this is based mainly on the short duration of dosing, but also the good monitoring and control in clinical trials. And if you want to read more about this, I've included the reference of Harvey et al. 2017 in the further reading at the end. But this is basically that when you're dosing a molecule for less than lifetime, toxicity is related to both the dose and duration. And if you're only dosing for a short period, maybe 1 day or 14 days or whatever, you can accept higher thresholds. And here, as an example, are the qualification thresholds that we apply in DNDi. I'd just like you to focus on the qualification thresholds because we don't have time to go through everything. But here you can see for drug substances, we apply a qualification threshold of 0.2% for unidentified impurities and 5.5 for identified. And on the drug product, during these early stages, we apply 0.5% for impurities in drug products. Now, from phase 2B onwards, we apply ICH. And just by comparison, the qualification threshold for an unidentified impurity in an API is 0.10%. And for a similar situation in a drug product, it's 0.2%. So you can see that we're using higher qualification thresholds here. A quick note about how you actually identify impurities. Well, in phase 1 and 2A, often you've only got small quantities of samples, etc. So typically you might use liquid chromatography mass spectroscopy, or you might isolate small quantities and analyze them using spectroscopic techniques. In the later stages of development, by contrast, normally you confirm identity of impurities by independent synthesis or isolation of larger quantities, followed by full characterization and spiking experiments to confirm the peak assignments in your HPLC chromatograms. A quick note that the ICH guideline M7 talks about controlling mutagenic impurities, and it's actually one of the few ICH guidelines that actually applies both at registration and during the clinical phases. And actually, there are some very specific requirements in this guideline for phase 1 clinical trials up to 14 days. So class 1 impurities, these are known human or animal carcinogens, need to be controlled to a compound specific limit. And class 2 impurities need also to be controlled to compound specific limits, or there's also some acceptable intakes in the guideline itself. Class 2 impurities are known mutagens, but their carcinogenic potential is not known. All other impurities at this stage can be treated as non-mutagenic and controlled to the normal limits. But also note in this guideline that there is the possibility to justify that an impurity is controlled at an earlier stage of the manufacture and to avoid the need for end product testing. One thing you'll see a lot in the headlines, in fact, there's a headline today about a Pfizer drug being recalled due to nitrosamine impurities. These are specific potential carcinogens that have been the focus of a lot of regulatory scrutiny recently. Whenever you have nitrosating agents and secondary or tertiary amines or their precursors in a chemical synthesis, there is the potential to form these. And so what you need to do at all stages of development is do a risk assessment of the synthesis, but also think about some other potential support sources. And it's been discovered on developing these guidelines that nitrosamines can also be found in contaminated starting materials, or if you're recycling solvents or catalysts, and certain packaging materials. And where you identify a possible risk, you really need some very sensitive methods to determine these. Just to give you an example, in the EMA guidelines, the permitted daily exposure of typical nitrosamines is between 24 and 96 nanograms per day. So you really need to use very sensitive techniques like LC with tandem mass spectrometry to measure these. I'd like to turn very quickly to analytical method validation. The extent of validation also differs by phase, and it can be broadly categorized in 2 stages. Stage 1, which is where we are in phase 1, products, processes not fully developed, knowledge is evolving, methods are used in a single laboratory, and this is an appropriate level of validation for phase 1 and 2A trials. Typically, with a method, you would look at accuracy, repeatability, specificity, limits of detection and quantification, linearity and range. Because it's only being used in a single laboratory, maybe method robustness is not so important, but you might look at things like the sample extraction procedures, the stability of the analytes, filter recovery, and this would all be done as part of the method development. Often at this stage, we're using a single HPLC method for API and drug products, so we can also combine the validation studies. Stage 2 is applied when we have the intended commercial product and manufacturing process. Registration specifications have been set, and we've actually got some well-characterized reference standards. And this is appropriate for test methods used for phase 2B trials and onwards. It really represents an enhanced level of validation suitable to support method transfer and product registration. And classically, this is all the parameters used in stage 1, but maybe we would look at intermediate precision. And this is the effect of maybe the instrument or the operator or the day, but still within a single laboratory. And we would also extend the robustness studies. So perhaps we would look at the deliberate variation of some of the HPLC variables if we're using an HPLC method. Finally, and I'm on the last leg now, I'd like just to turn towards some differences in stability assessment, which is a very important topic. Well, first of all, it's fair to say that regulatory expectations are pretty similar at all stages of development. Regulators expect you to demonstrate that your investigational product remains within acceptable physical and chemical limits for the duration of the clinical trial. They expect to see in-use stability, perhaps after you've reconstituted or first opened an investigational product. They want you to think about other characteristics or quality attributes, microbiological, perhaps you're using a non-preserved formulation, for example, or interactions with administration sets, things like infusion lines, syringes, dosing cups, etc. They expect you to perform purposeful degradation studies early on and to use stability indicating test methods. I think that should be obvious. And obviously, in a clinical trial application, you have to justify your storage conditions and your shelf life from the data that you've gathered, either on your investigational product itself or maybe some closely related formulations. Again, stability assessment is a holistic activity. Normally, we perform purposeful degradation studies looking at the effect of temperature, humidity, acids, bases, oxidants, light on our molecule. We aim to get 5 to 10% degradation typically in these studies. And it's useful because it helps identify potential degradation products as well as show that your methods are stability indicating. We do pre-formulation studies. That's really to demonstrate that you've chosen an acceptable form of the API and that the excipients are compatible with it. We've talked already about qualification of impurities, so I won't discuss that again. But also, you know, again, we may have some experience at this stage from batch release data or our stability studies. But as you can gather at this stage of development, these data sets are very limited. One of the big differences between phase 1 and later stages of development is that extemporaneous preparation or compounding is used very frequently within the industry. And extemporaneous preparations, well, you still have to perform long-term and accelerated studies on the API or a drug product intermediate if you're providing one. But in extemporaneous preparation, the doses are reconstituted according to a pharmacy manual and administered immediately. And so for reasons of practical convenience, typically we would do in-use stability studies. They don't really need to be longer than 24 hours. Depending on the phase 1 unit you're working in, the conditions could be refrigerated, ambient, or even under controlled room temperature. And typical tests are appearance, assay or drug content, pH, and impurity monitoring. But also for suspensions, you might also consider uniformity and dissolution. Well, shelf life assignment, the problem at the start of phase 1 is that you have very limited data available. And in this case, it's quite typical to assign a default shelf life of 6 months based on existing data. You've already got extensive API stability data, pre-formulation studies, and you've probably also put some prototype formulations on stability. Regulators expect you to put a shelf life extension plan in the clinical trial application so that you can extend the shelf life without having to keep revising your clinical trial application. And they also expect a commitment to maintain the stability study to cover the duration of the clinical trial. And obviously, if the stability studies show some negative or adverse trends, they also expect you to inform them immediately. One topic which is often a little bit confusing is the type and number of batches that you should put on stability. And actually, at this stage of development, you can use a technical batch as a stability batch, as long as its quality represents the eventual quality of the clinical batch. And often a technical batch is placed on stability in advance of manufacturing the clinical batch to facilitate smooth expiry dating and no interruption of clinical supply. There's actually no need for 3 stability batches in early development, although often companies will place a technical batch on stability to begin with and then a clinical batch afterwards. But just remember, for some formulations, you may never make more than 1 batch. So clearly it's not practical to make 3 batches. In terms of stability protocols, very quickly, typically we use the ICH conditions and test schedules in our stability protocols. The long-term condition that we choose depends on where the phase 1 study is being conducted. For climatic zone 1 or 2 regions, 2560 is sufficient. But there are some alternative conditions on this slide. For example, if you're working in a climate zone 4 country, then 30 degrees, 65% RH or 3075 may be appropriate. And also we have accelerated conditions. Concerning the shelf life extension plan, well, actually in the EMA guideline, it actually gives you some rules for doing this, which are quite easy to follow. It allows a 4-fold extrapolation of your accelerated stability data up to a maximum shelf life of 12 months. But from 6 months onwards, they will allow you to extrapolate 12 months from the amount of long-term stability data that you have. And again, this is my final slide before the summary. This is actually the shelf life extension plan that we routinely use in DNDi, and if you have the slides afterwards, you can obviously look at that. So in summary, products and processes are not fully developed at phase 1, and this is, I hope you understood the concept of learn versus confirm. Of course, we can anticipate ICH requirements later on in registration, and this is what Ready by Phase 3 is about, but we can't really apply ICH guidelines fully at these early stages. So we have to adapt our quality requirements and practices for phase 1, particularly in respect of impurities, analytical methods and validation, and stability assessment. And these practices are generally accepted by regulatory authorities that frequently review clinical trials at this stage. And together with GMP, obviously these things assure the quality of the investigational products used in the trials. So thank you for your attention. There's some further reading in the slides, and I think this was also provided separately as well, if you want to explore any of the topics in more detail. Thank you again, and happy to take any questions that you may have. Thank you.
Thank you, Dr. Stephen. I think so it was a very interesting topic and a lecture talking about the various quality aspects. There are a few questions which have been asked, maybe like if we can answer them. There was one question regarding the phospholipid, and the participant wanted to understand whether phospholipid, especially the phospholipid of choline type, can produce any nitrosamines.
I'm not aware of any specific risks around phospholipids. I imagine, are we talking about these as use in maybe excipients? I'm not aware of any specific risks, but again, my advice would be do a risk assessment or contact the supplier and ask if they've done a nitrosamine risk assessment. Not just on on the manufacturing process, but also the materials like solvents, etc., used in the manufacturing process.
Thank you, Doctor. Yes, this was indeed a question with respect to excipients and use of phospholipids in the excipients, and I think so. You have very well said is that it would be better to do a risk assessment, connect with the excipient manufacturer to get more information, and probably then do an evaluation of the risk. There was one more question which was asked, was with respect to the impurity calculation. I also could not get that properly, but the question was like, what weight is considered for impurity calculations? If you can.
What weight?
Yeah.
So in this calculation, if you look in the formula, basically you've got the no observed adverse effect level, which is always expressed as milligrams per kilogram per day. This will come from the design of your tox study. You have the impurity level actually in the batch, which is expressed as a percentage. And you have the body weight for a human, which typically depends how conservative you want to be, but typically people use 60 kgs for the human body weight. And then the expected maximum daily dose is again expressed for humans is expressed in milligrams per day. And if you get the slides afterwards, you can see the formula. But if you use those units in the formula, you'll end up with the qualified level in percentage, which will give you directly a limit to put in your specification.
Thank you, Dr. Steve. I think so that answers the question. And as you rightly said, that the presentation would also give the calculation, which would be helpful to do this calculation. So I think so we are close to the closing time, and I would like to thank Dr. Ravinder, Dr. Vivek, Dr. Steve for a very good knowledge and experience sharing. Your presentations explained how important the role of drug substance characterization in formulation development is. We also understood that in the early development, how we can accelerate the formulation strategies and mitigate risk. Today we also touched on some of the tools which are available for to accelerate the development so that we can make formulations available early for the clinical studies. Dr. Steve also talked about the quality attributes for the phase 1 formulations and the regulatory requirement. In overall, the presentation touched on various important aspects of formulation development, which is a very critical aspect of any dosage form, making it available for the market. All the formulations, in fact, probably I believe that the participants must have got benefited hearing to them. I thank all the presenters for such an informative talk. I would also like to thank the organizers, DNDi, BIRAC Regional Biocenter, Innovation Biocenters, then Indian Society of Clinical Research for organizing this very important workshop. I thank Dr. Maushmi and all my workshop organizing colleagues for their support to make this day 2 session quite interesting and successful. One note to all the participants is that the feedback form link is available in the chat box at the end of the webinar. This link will be active for next 10 minutes. Kindly fill your feedback. Thank you very much. I hand over to Dr. Maushmi to conclude.
Thank you, Amit. On behalf of the organizers, I take the opportunity to thank all 3 speakers and our session chair for a very informative session. We also appreciate the presence of 270 plus participants today. Thank you for your time and interest. We look forward to seeing many of you over next 3 days. I also request that our session chair and speakers to turn on your camera for a photo session. And I would also request Akhila to share the poster of today's program on the screen for a photo.
Yes,
Thank you. Okay, give me a second. I will turn off my video and take a photo. Thank you, everybody. Have a good evening, and for others who are on the West, have a good rest of the day. Thank you.
Bye-bye.
Thank you all. Bye-bye.
Thank you. Bye.